Part 2. Customized response to disease and finding drug targets. Drug responses are remarkably complicated. Drug efficacy may be affected by all sorts of things: transporters that move the drugs around, metabolism, receptors, signaling pathways, and so on. And all of these things can be affected by mutations, because these will change how effective proteins are at transporting, metabolizing, and so on. Many drugs are actually metabolized into an active form, and also many drugs are metabolized into an inactive form before being excreted. And one particular protein, cytochrome P450, is involved in many of these cases. And that means that people may be poor, typical, or ultra-rapid metabolizers, either in metabolizing a so-called prodrug into an active form, or metabolizing drugs into inactive forms for excretion. There are more than 40 well-known examples of drugs to which different people have very different responses, and these include things like AZT, antiarrhythmics, antidepressants, antipsychotics, and opioid painkillers. So, for example, codeine is actually a prodrug, and more than 2% of the world's population is homozygous for null alleles that mean that they cannot metabolize codeine into the active painkiller. Now, as we've seen, different populations can have different occurrence of particular alleles, and consequently drug responses can be dependent on the population. So mefenitoin is a drug that's used for epilepsy, and around about 2 to 3% of Caucasians are poor metabolizers. However, 23% of Asians are poor metabolizers, most commonly one particular mutation of G681-A, which causes aberrant splicing. And therefore we can have population-specific recommended doses of drugs. Now, drug responses are in fact polygenic, so as I've mentioned, things like uptake, metabolism, and responsiveness are all going to be related to effectiveness of the drug, and clearance, which of course also includes metabolism, is going to be related to toxicity. And not everything is related to the genetics. So, for example, there was an antihistamine that was very popular some years ago called Triludan, and the problem with Triludan was that some people started dropping dead. Now, the reason for this ended up being that they were eating grapefruit for breakfast or drinking grapefruit juice. And the problem there is that grapefruit is well known to contain an inhibitor for cytochrome P450. So they were taking a standard dose of Triludan, and the Triludan was not being broken down and therefore not cleared from the body properly and built up to toxic levels. Finding drug targets. So let's look at a scenario. A new virus appears. So when I first created this slide, this was SARS back in 2003. But it could be something like Zika or SARS-CoV-2, of course, at the moment. Taking a kind of bioinformatics approach to understanding what's going on, then the first thing we want to do is sequence its genome. So SARS in 2003 was done in just under 2 weeks. SARS-2, of course, was done in under a day. Once they had the isolated virus, they could sequence it very, very quickly. Once we've done that, we can easily screen for similarities with other viruses, and we can derive protein sequences. Now we can also quite easily look for related proteins of known structure if those exist. So it's easy if they're there, it's not so easy if they're not. And from that information we can transfer annotations of likely gene function. So that might be at a structural level or just at a general sequence level. We can build models or try to do ab initio modelling, which isn't really possible yet on a really reliable basis. And we can use that information then to help us with the design of antiviral drugs and in fine-tuning and modifying antibody-based drugs. Of course, with SARS-CoV-2, as I mentioned at the beginning, having this ability to do sequencing really fast and remarkably cheaply means that people's infections can be sequenced very quickly, very rapidly, very routinely, and that can allow us to track changes in the virus and how the virus spreads through a population. Of course, in that case, because we also have structural information about the SARS-2 or SARS-CoV-2 virus now, we can also map those mutations onto the structures and understand how those may have effects, for example, in being more infectious. So, for example, the Kent variant has mutations in the receptor binding domain of the spike protein where it interacts with the ACE2 receptor, and that allows us to see how these mutations can be correlated with the more infectious nature of that variant, because it seems that it binds more strongly to the ACE2 receptor. Similarly, we can look at other variants as they come up in a structural context and try to predict whether they're likely to be variants of interest. So how could we use this sort of information in order to identify drug targets? Well, in the case of the SARS-CoV-2 virus, the most obvious target is the spike protein, because that interacts with the ACE2 receptor. In the general case of anti-infectives, we're looking for targets which have no close human homologues. Now, one of the arguments that has been presented by some of the anti-vaxxers in the case of the COVID-19 vaccines is that the spike protein has homology with a protein called syncytin, which is involved in pregnancy. And therefore they're suggesting that having the vaccine may lead to infertility in women. Now, it is indeed true that syncytin is probably homologous to a region of the spike protein, but it's only under 18% sequence identity. Now, given that we're concerned about mutations in the spike protein, meaning that antibodies that have been raised against the vaccine may result in the vaccine antibodies being ineffective, and there we're talking about a couple of mutations over the size of the spike protein. So we're talking about probably more than 98% sequence identity being something of concern. When we've got under 18% sequence identity, the chance of an antibody against the spike protein cross-reacting with syncytin is virtually zero. However, in general, when producing anti-infective drugs, what we're looking for is targets that have no close human homologues in order to avoid any chance of a cross-reaction. If the drug that we're looking to design is against a host protein, then ideally we would look for targets with no paralogs, again in order to avoid off-target side effects. In reality, around about a third of small drug molecules actually all interact with G protein coupled receptors, so those are all one big family. But this is something that people are kind of aware of and trying to choose targets and looking for homology, and then looking at the regions where the drug's going to bind are things that are incredibly important in making sure that your drug is not going to have major side effects. So the sorts of things that we need to do then are to annotate sequences, look for homologs with known inhibitors, look for homologs with known structures so we can build models as a kind of precursor really to going on to solving structures, looking at the sequence to predict things like location, post-translational modifications, and so on. A term that you may come across when looking at design of drugs and identification of drug targets is drugability. And overall, this tends to be defined as how well a therapeutic can access the target, but it's also defined as the efficacy that a therapeutic can actually achieve. So parameters that influence drugability include the cellular location of the target, the possibility of the development of resistance against a drug, transport mechanisms such as export pumps, side effects and toxicity. Using known successful target classes such as the GPCRs that I just mentioned does rather sidestep that problem because we know that these are druggable targets. Drugability can also refer to the drug itself, the idea of drug-likeness, in other words, compounds that are likely to yield useful leads for producing drugs. And there's a set of rules known as Lipinski's Rule of 5, which gives a number of rules that can be considered to look at how drug-like a particular small molecule is. Now, of course, there are always exceptions. There are a number of drugs that don't really fall within the requirements of Lipinski's rules, but nonetheless, if you're screening a big library of potential molecules, selecting ones that do fall within those rules is always a good starting point. We're going to take another break at that point, and in part 3 we're going to come back to mutations and analysing the structural effects of mutations, and also looking at introducing mutations to engineer proteins for use as therapeutics.